want just in the beginning to say that most of the data that we are using to do this kind of analysis derive from teamwork, basically based on two efforts. One is a time series that we are in Naples, and this is only people involved a lot, and the Tara Ocean. Probably you might have heard of the Tara Ocean. So,
you should not stay in front.
the strange.
Maybe you can.
So the disclaimers, as seen. First of all, this is not a view of a theoretician, but a reader of end user. I'm not a theoretical physicist working on networks, but we are just starting to use networks. And what I will show today is more mechanistic deductions than holistic and topological. The reason is that plankton webs, as other webs, have been highly simplified so far and have been essentially dealt with for trophic interactions. So this, basically the idea is that the edges of the network are mostly trophic. And I have to say that the truly network approach is still in its infancy. But there is some tradition of individual-based models, object models, in plankton interactions. So in the scope of this presentation is to present a case study to show what are the gaps that are quite relevant, and also ask for feedback from kind of suggestions. And so first of all, what is plankton? There is this traditional division of phytoplankton and zooplankton. Phytoplankton are the autotrophs, and zooplankton are the consumers and the grazers, and the size range is between 0.6 to 1,000 microns for phytoplankton, and between 20 and 100,000 microns for zooplankton, so they are small. This kind of very schematic division takes out heterotrophic bacteria, viruses, and parasites that are becoming more and more known, and also whose role is becoming more and more important. Just to frame the importance of plankton on the Earth system, this is a recent estimate of global biomass. It's a very good work that has been published last year on PNS. And maybe the global estimate of how much biomass is on the Earth. And if you look at the differences, on the Earth there is 550 petagrams of carbon, whereas in the marine system only 6 petagrams. But the big contributors of the biomass on the Earth are the structural parts of the plants, so trunks, etc. So the imbalance is not so strong, but still, biomass, marine biomass, is much lower than terrestrial biomass. And if you look, if you focus on the role of plankton, this is something quite new, interesting. That if you take the estimate of the biomass above the sea bottom, so without considering what is in the sediments, the plankton, which is the organism of the size classes that I was showing, make up 80% of pelagic biomass. So forget about whales, sharks, cods, etc. The largest part of biomass in marine systems is macroscopic. So this is one important message. And if you consider also the plants and the animals living on the benthic layer in the coastal regions, on thermal vents or seeps, then still plankton makes 60% of marine biomass. Okay. Then why they are so? How do they make this biomass? Because they are very numerous. These are the numbers. Heterotrophic bacteria account for 10 to 28, 29 cells. Prochlorophytes, which are the smallest cyanobacteria, phototrophic bacteria, 10 to 27. Photosynthetic epicoacariotes, 10 to 24. They are a lot. They are very abundant. And if you consider, if you normalize for a unit volume, then we have 10 to the third to 10 to the 7 unicellular organisms per cubic centimeters. So in a milliliter, you have this kind of density. This is lower than the density you have in the soil, 1 or 2 other magnitudes, but still you have so many organisms that this would be, in theory, a very complex system because you have many, many agents that are potentially interacting. So, even in terms of rates and of biological activity, if you take the scaling law of Kleiber or Brown, the metabolic theory, etc., and you assume a very simplistic power law, and then you consider the biomass ratio between the small planters and the large organisms, then you come out with the estimate that plankton rewards million times more material than the other animals and the other organisms in the marine environment. So this is for this reason the plankton has a key role in most of the relevant biochemical cycles of the Earth. First of all, the carbon pump is 50% operated by plankton in the ocean. So this gives you the relevance of this component of biota. And then we have so many organisms, we say that they are quite, the density per milliliter is quite high, but then comes the other problem. If you assume that they have a random distribution, and then you take the average distance among the planters, among the cells, then you come out with the separation, average separation lambda, that goes from 50 microns to 1600, which means from 1 to 3 cell diameters to 10 to 50 cell diameters. So they are to some extent diluted. So on average, the interaction distance for poorly motile organisms spans from 25 to 50 cell lengths to 31 to 50 cell lengths. Where cell lengths is the cell diameter. So this is the picture. This is a very extreme environment in the tropical areas where the abundance of these organisms is quite low. And this is a prochlorophyte, the smallest autotrophic organism, and you can see from this picture which are the crucial scales. And for example, the cell-to-cell distance is 200 cell diameter. So they are in a random distribution, they are quite far one from the other, despite their total abundance. So for this reason, quite recently has been rescued the concept of the phycosphere, which is the area, the volume around the cell that is affected by the cell activity, and is the volume that in which you can have interactions or uptake of nutrients or release of substance, signaling molecules, etc. And this deformation at different Peclet number is the number that states that is an dimensional number that estimates the ratio between advective flux and diffusive flux. So is the ratio between the chemical diffusion and the fluid motion because of the fluid dynamics. So, and the phycosphere concept is that there are microdomains where external disturbances after a long scale that because then diffusive processes, which means that the turbulence is not strong, it cannot affect for immediately the thickness of the volume of this layer. So substances that are in this phycosphere can stay enough long to become signaling molecules or whatever. And this is a sort of a general picture. Of course, it depends on cell diameter, so you have different cell sizes. You can estimate that in the phycosphere, you can consider the phycosphere, which is the cell boundary layer, something that is with concentration of a given substance, 1 to 0.5 in respect to background condition. And then you have a range of possibility. Here is the lower detection bacteria, and these lines show the effect of turbulence in distorting and squeezing and reducing the phycosphere. And we know several possibilities by which bacteria can access to the phycosphere. There is Brownian motion, random encounter with the cell that swim, chemiotaxis, bacteria are quite known for chemiotaxis, and finally, probably the most effective one is the direct action cells. So but still, the encounter rate, if you is just Brownian motion, is in the order of 0.0035 cells per day. So it's quite rare, the encounter. It goes up to 9 phytoplankton cells per day, and then with the chemiotaxis further increase, the attachment, of course, you have a pattern. So the second message is that they are quite diluted, and then the interactions are not so frequent. However, we know that there are interactions in the phycosphere scale. They are studied mostly in the lab, but we know, for example, synergetic interactions with some bacteria provide vitamin B12 to phytoplankton cells, because not all the phytoplankton cells can synthesize vitamin B12. There are exchanging nutrients like ammonium for nitrogen fixers that can provide nutrients to nitrogen to cells. Or even there are symbions that can do this. There is exchange of the solid organic matter. There is some gene transfer, which is a different mechanism, but a lateral gene transfer from bacteria to eukaryotes. Then there is also some parasitic attachment. So we know that there is these mechanisms are active, and we even know that there is some relationship. It is not just trophic, but is also informational, because this interaction is between a diatom and a bacterium, in fact, because exchanging signaling molecules is such that the bacteria gets some organic material from the cell, and in fact, via indole thiazenic acid, induces the cell division. So it's not just a trophic interaction, it's a regulative interaction. So bacteria can affect the division rate of the cell. We also know that there are several interactions among protists. This is a network, I'm showing the network, but this is a study that has collected all the known interactions among protists, and there are prey-predator interactions that are most abundant for different groups of different classes, and also parasite toss, symbiont toss, and interactions that we don't know, but they are quite common. And this is the degree, how many, typical the degree of networks, how many, this is a log scale, this for predators and hosts, and this for prey and parasite symbionts interactions. So with how many organisms one belonging to this class interact with the other organisms, and this is the specialization. So for example, parasites, symbionts in many interactions are highly specialized, meaning that they can have only one or two targets. So again, we have the evidence that there is a possibility of interacting, but we are still missing the picture of how it can work, because they are diluted, and I will show another problem that we are facing. So if you take as a relevant scale the ratio between the phycosphere radius, which is the area, the volume that is affected and can act as a coupler with the other organisms, versus the average distance, we see that goes from larger than 2 to 10. So the phycosphere is smaller than the average distance among the organisms. And if you take the time scales, biological time versus the time to diffuse from this distance, then the biological time scale is in the order of fractional day, say the division rate, and tau is in the order of seconds. So, if you go out from the phycosphere, diffusing from the phycosphere, you can, in theory, send signals with enough speed to couple with the biological time, physiological time of the organism. But the opposite problem is that the marine water is not steady, is in continuous mixing because of turbulence at different levels. And so the turbulence displays water parcels one respect to the other. And if you take a quite low level of turbulence with the distribution, kinetic distribution rate of in the order of 10 to the minus 6, then the shear rate, which is the sliding of one layer of water with the other, makes that the relative velocity of one particle to the other is in the order of 11 microns per second. So this means that if the water is not absolutely calm, the organism has very little time to interact. So this is this means that the apparent space-time of interaction is in the order of fraction of hours for medium-sized cells. So in synthesis, the problem that we are facing is that there are established interactions that we observe in the lab. We know the molecular mechanisms, but from what we get from our knowledge of how the logic system works, the interactions apparently should be very transient and ephemeral. I mean, you have a little time to interact efficiently. And for example, the one thing that I was showing about the mutualism between the sulfobacteria and the diatom, in the experiment carried in the lab showed that the effect of regulation of cell cycle in the lab because of acetic transmitter was the effect was resulted after 14 days staying in the same culture. So this is quite difficult to reconcile with the fact that cells move relatively one to the other, unless, and this is one hypothesis that we work, that there is an effective niche construction, meaning that they are structuring the environment around the cells so that what we compute in terms of relative displacement is not effective because they make exudates, they structure the fluid in a way to prevent relative movement as it would be in a clearly random walk thing, like a completely unstructured fluid. So this is one possibility. And also, again, if we move up in macroscopic patterns, we see that there are very strong temporal patterns. The succession of plankton, phytoplankton in this case, is very well established. You have species that succeed one after the other. This is the result of 20 years of data in one site. This is a time series, and there are these species, and you can see that there is a clear time course in the succession of the species. So the message that comes from here is you have quite a well-organized system in time, and also in space. These are the data, and in space you can construct a biogeography These are metagenomic data, so it's quite well resolved because they are made using the metagenomes of samples and making a distance among them. And you can see that there are clear areas of evolution that display common and similar communities and are different from other communities. So there is also an evident structure in space. So now we go to the finally we have to the web networks. I cannot give an important contribution from a theoretical point of view, or because we are just beginning to use this. So normally we do our concurrence networks. We just use data to look at when species, or genotypes co-occur together. And these are two data. These are Tana data, and the way in which we ordered the network was using the weighted gene co-expression network analysis, which is one of the techniques that is used, but used in gene networks to highlight the genes that have the same pattern or that are expressed together in a coordinated way. So we use that technique to look at different clusters of community, and we use the same similar kind of approach for the Gulf of Naples in our time series. And what we got was that we have this time series in summertime. When we sample in summertime, there is the advantage of being able to sample the same community because there is recirculation inside the bay. So sampling the same community, we got two different structures of the same composition, but with different abundances. So we could separate what we call the green network and the blue network. Blue was for low trophic regime, and green was for high trophic regime, where a trophic regime is defined as the amount of phytoplankton biomass, so eutrophic on oligotrophic. And we made this kind of networks with different nodes. Sometimes the nodes are species, when we could resolve the species. Some other times are grouped together, like some heterotrophic bacteria are all together. Okay? And what we found, and this is what links to what Sadiq was saying yesterday about the switching. This is what we observed, that if you move from the green state, the trophic state, to blue state, or the trophic state, we observed the switching in the edges, and these switches was due to the way in which the carbon would flow in the food web, because in one case, when you have a trophic states, the grazers, the zooplankton, is mostly grazing on the phytoplankton, and protists are feeding on phytoplankton. But when you have the oligotrophic, so-called oligotrophic state, then zooplankton feeds on protists, and protists feed on phytoplankton. This has a relevant implication in the carbon transfer up to the food web, because the system is displaying a nonlinear response in terms of carbon transfer, which means that if you double the primary reduction, you don't double the carbon transfer upward in the food web. Normally, you have, if you increase 7 times the primary reduction, the expert to higher level of trophic will be only 3. So you have a lot of recirculation within the unicellular component of the community. And then to go more in depth in this, we try to weight the network, to produce a weighted network. And to do this, we found out the different trophic links among the different groups, metazoans, unicellular in the surface and in the intermediate layer, and to be sure that this was enough reliable and robust, we used a model that is called EcoPath, that is regularly used in fishery, which is a mass balance model. So assuming the steady state, we use it, and having the mass of all the nodes, we iterated simulations until we found the carbon transfer among the different nodes that would satisfy the steady state. And doing this, we could weight the network. So we have a weighted network, and we built again the weighted network, which is this, and we are just starting to run some analysis, like indirect weight effect and weighted centrality, etc. And again, we confirm the fact that the green and the blue state are structurally different. So there is a rewiring of the network, just changing trophic state, and the same kind of thing we have found at the global scale with Taradita, where the same community is modular, is divided in modules, and the modules respond differently according to the change in the environment. So for example, we ran an experiment on Maltese's islands in Tara, and when you have a natural hydrofertilization because of the presence of the islands, there is the island weight effect and other mechanisms, physical mechanisms, we found that in the same community, different modules respond differently. Same network with modular structure, some modules respond positively, some other modules respond negatively, which means that there is also there a rewiring of the network. So this is something that we are working on, and any kind of suggestion would be very appreciated. And then the last thing that I want to show is this is microscape, and if we move at okay, this is what I was saying before the switching among the modules. You see, we use infomap to determine the modules, and we observe that in the transition you have a species or group of organisms that switch module. They go from one module to the other, as is shown here and here, and which of course has an effect on the carbon transfer. And finally, I want to show at the microscale another problem that we are facing. Okay, this is a typical it's called spaghetti diagram, which are we normally use a technique to track the currents, and you put drifters, floating Newtonally buoyant drifters that send the position to the satellite. So this is the Gulf Stream, which is quite known. But you see that even in the Gulf Stream, which is very consistent and high volume water flow, you have all these variations from the trajectories. So from the Gulf Stream, you can go down. You don't go to England, but you go to the subtropical Atlantic Ocean. So this is what plankton is subjected to. So besides the relative displacement at the microscale, you have the fact that the community that starts here, part of that can be dispersed. So there is a lot of dispersion that is related to the transport in the ocean. So we had, having this picture in mind, we took the Tara data and with the Lagrangian model, with tracking the particles, we computed the most relevant tracks. Was not so we just took the minimum time to go from one station to the other, and we tried to see which was the ratio between the minimum time and the composition of the community, the travel time and the composition of the community. And we found another very interesting pattern that you have up to 1.5 year, you have a memory of the transfer. So plant cells are transported by the flow, they are dispersed, so you are losing the original composition. So the beta diversity is decreasing in space-time, because the key message that they want to give you is that plankton is different from any other system on the Earth because it has no substrate. It is in a fluid medium which is continuously moving, so you cannot map on a specific place. You know, even birds fly, insects fly, but then when they have to reproduce, they have a substrate. They stay on a tree, they stay on the soil, etc. Plankton has no fixed place. Despite this, you have this emergent pattern where you have that the distribution is quite structured in time and space. So now, if I want to wrap up what are the open problems, we have knowledge gaps on type of interactions. We don't know, we know some mutualistic interaction, we know some symbiotic interaction, but we are missing the complete picture because we have knowledge gaps in the biology of the players. For example, there is we don't know how important is mixotropy. Mixotropy is another thing that is quite present in marine systems, whereas not so common on the Earth, on the solid Earth. And we don't know too much about life cycles, which are probably the system that organisms use to synchronize their presence or absence from the environment, despite the fact that they are moved around. We don't know how the information is transferred among the organisms, because it can be optical, but for example, copepods don't see. They don't have an eye, but they have very, very refined mechanical sensors. So probably the mechanical sound, say in wide sense, which is transmitted very well in marine environment, can act. Chemicals, we know that there are pheromones, but still, when you have pheromones, there is a diffusion problem and the problem that the water moves. So you cannot, this problem is common also in the atmosphere. Insects have the problem of the wind. But pheromones have been detected for sexual reproduction. So we know that there are a lot of chemical signals. We don't know about electromagnetic, being the medium, a lot of ions. We don't know if there are local current that can act as an information transfer. So again, I wanted to connect what you were saying yesterday, that we are missing how the information is transferred, not just the matter. The plankton ecology has been a lot worried about how the matter is transferred, how carbon, so there are many food webs, but we need too little about information webs. So how the information is exchanged. So the only conclusion is that the perception that we have that these communities have high plasticity just because they don't live always with the same mates. So they are continuously changing the composition. So even if they probably can engineer their local environment, still there are a lot of forces that tend to separate them, and so we have these contradictory views. On one hand, we know that they interact and they can even regulate the life cycle of other organisms, but then we have on the other side the fact that they are continuously moving one versus the other. So is the problem the time scale is apparently too short. And the reason why this gap of knowledge is obvious. One is because they live in marine environment and we don't have a direct observation. And now we are improving with new technologies like bioptics and acoustics and molecular tools. But one thing is that you're in a forest and you see the animals and they're moving, and one other thing is that you go on the ship and the water is below. And the second reason is because they are microscopic. So, and because they are microscopic, it's very difficult to observe their behavior in situ, and this is something that introduces a big bias, because all what we know about their biology is produced in the lab, and the lab conditions are very, very different from in situ conditions. So in the lab we can detect which are the ranges, what they can do. But we don't know if they are really doing this, or if what we see in the lab is a dominant response inside it. So this is a gap that we have to fill. And going back to the networks. So we have evidence from the plankton was quite straightforward to have this picture of rewiring of the webs. So they are very dynamic. And this was something that came out in discussion. So this is quite evident, even from a few data. But we are still missing, besides the carbon transfer, that it's just an inference, because it's just a mass balance. But we don't know if it's really going into it. We tried also Monte Carlo method to see if that structure was the optimal one, and came out to be quite robust. But we don't know the edges. We should work with multi-layer networks, which, but in the different layers than just carbon transfer, how can we characterize the edges? So this is what the next thing that we would like to do. Okay.